go ahead and do that. I'm Bruce Korf from the Department of Genetics, and was asked to talk this hour about the broad topic of genomic medicine. I think you'll be hearing from somebody in the second hour about some of the computational aspects of genomic or precision medicine. And let me begin with a general statement about the concept of precision medicine and really the notion of individuality. You hear a lot about these various terms: personalized medicine, precision medicine, go on and on. I conceptualize this really as using data about an individual to guide medical decision-making, prevention, diagnosis, and treatment. And if you think what constitutes individuality, it's a lot of things. And this is not meant to be an exhaustive list. It includes ancestry, and I mean by that really, in a sense, genetics. And that's what we'll mainly focus on today. But it's not limited to that. Every person has grown up in and lives in an environment that is collectively unique to them and would involve various kinds of exposures, for example, that might be, the sum total at least, different for every person. Cultural issues will factor in here. How people view the very concept of health and disease will be an individual kind of thing. And finally, personal experience, the degree to which an individual has had experience with a particular condition in themselves or in their family, and how they look at the issues of prevention and management. These are all individual things. And it isn't new to be thinking about the individual. I would argue that's what medicine has always tried to do to one extent or another. What is new are the tools that we have at our disposal to enable us to mobilize these various bits of information. And you'll hear later about some of the informatics tools. I'll focus mainly on what essentially is the issue of ancestry, but really we'll place it in the context of genomics. And all of this really converges on the ability to identify individuals who are at risk of disease, in the hope that, knowing who those individuals are, you can customize a plan to avoid the eventual development of disease. Those who end up needing treatment, you hope to be able to monitor their response to treatment and to optimize that so that there are fewer side effects and better outcomes, and really to be customizing choice of medication to the individual. So I want to talk in general terms about a number of areas that specifically relate to the component of individualization of care that is genetic or genomic. And we'll cover the things that are listed here. I'm going to start with rare disease, which, although by definition, well, rare, but actually collectively rare disease is said to affect about 10% of the population. So maybe not so rare when you think of it that way, but these are the, I guess you could say, traditional domains of single gene Mendelian sort of medical genetics, but a lot has gone on there, and I think it's worth a few minutes to give you a sense of what's happening. The inverse of that, common disease, has also been the subject of a lot of advance, especially even in this past year or so, and you're going to start seeing that filter down into the clinic probably faster than you would imagine. Thirdly is cancer, which is really fundamentally a genetic disorder. I'll say a few words about epigenetics and then give you a sense of some of the things happening here that you can get involved in if you're interested. Key here. So a lot of people, when they think of genetics, think about this sort of scenario. The picture in the upper left is a karyotype that was produced in the early 1960s, about when cytogenetics was sort of born as a field. The child to the right you'll recognize as an individual with Down syndrome. And indeed, there are, let's see if this pointer works, altogether 3 copies of chromosome 21. It should be a familiar kind of scenario, and for many people this is how they think of, relatively rare, in this case, genetic issues. This is a more modern view of how cytogenetics is done using a technology of cytogenomic microarray. In general, what is done, and there are a variety of technologies, but we now have microarrays that contain on them hundreds of thousands of what essentially are oligonucleotides representing unique regions of the genome. And you can query those as to whether there's a normal copy number for a particular patient sample or not. The exact technology isn't that important. You get pictures like this, and if there's normal copy number for any of the various probes, each of which is represented by a dot here, then it will kind of lie at the baseline way down here. Maybe you can see that there is a displacement from the baseline. It represents, in this case, a deletion of genetic material way too small to visualize with the light microscope, would never have been picked up. Certainly not with the technology I showed a moment ago, even with the more refined chromosomal analysis that began to be done in the 70s and 80s. But now, the example shown is autism spectrum disorder. The pickup rate for variants has risen just with this technology from somewhere in the 5% range to somewhere in the 20% range. Copy number variants that are of a few 100,000 base pairs of DNA that still contain multiple genes and can have a profound effect on development, but which would not be visualized if one used the old-fashioned technology. And this is really now the mainstay of what goes on even here in the cytogenetics laboratory, is the use of this microarray technology. But meanwhile, there has been a revolution in genomic sequencing. This is a graph maintained by the National Human Genome Research Institute. And it's based on their support of genome centers, and through that, they have a window on how much it costs to sequence a genome. And so this began in 2001, right around the time the human genome sequence was being completed. And at that time, notice the y-axis is a logarithmic axis, it cost about $100 million to sequence a genome. The first kind of completion of the human genome sequence collectively was thought to be somewhere around $1 billion. Well, in the course of what I guess are about 3 or 4 years here, it kind of gradually went down and followed Moore's law. You may be familiar with Moore's law. It's the idea that the density of information that you can put on a computer chip doubles, I think, every 18 months or so. That's how come the computers in your pocket are more powerful than the ones that would fill a room 30 years ago. And geneticists actually, as you watch this curve over the course of about 7 years, the curve of sequencing followed Moore's law pretty well, and it went down by a factor of 10. So in 2007, it was a $10 million exercise to get a genome sequence, which is still a little bit more than you probably could successfully bill to insurance if you were trying to do it clinically. But geneticists were kind of proud of the fact that they had tracked Moore's law, which has obviously been a very powerful kind of engine of advancement. But then you notice in 2007, the cost sort of fell off a cliff, and it just plummeted to the point now that this was last updated a couple of years ago, I guess. It had more or less reached the baseline of the $1,000 genome, which many people viewed as the kind of holy grail, that if you can do a genome for $1,000, it really would bring it into the realm of affordability. There was a catch to this, which is Moore's law has continued its trajectory, more or less. Here, genomics has way outpaced the trajectory, which you might think is a good thing, until you remember, what do you need to use to analyze genome sequences? And the answer is computers. And if the sequencing has outpaced computational capacity, which it sort of has, that it would create major problems. And it's been a challenge. The density of data that you get from a single genome sequence is too large to even send through the normal sort of cables that one uses to connect computers together, for example. Used to be said, actually, that we'd succeed in having a $1,000 genome coupled with $1 million interpretation. And glad to say that's not entirely true, that in fact the computational tools to analyze the data have advanced quite substantially too. So we're now in a pretty good position to do the analyses and to actually come up with diagnoses. Let me just show you a little bit about what's done. So you'll hear about the distinction between exome and genome sequencing. Let me just quickly give you a window on what we mean by that. One way or another, you start with genomic DNA. To do exome sequencing, you essentially create a library of fragments of the entire genome. But then what you do is you capture the exons, those are the coding sequences, you'll recall, the things that encode protein, by having a set of oligonucleotides corresponding with all the known exons. And so when you do that, you isolate just the DNA in this library that corresponds with protein encoding sequences, and that's what you end up sequencing. That's exome sequencing. So in principle, you're looking at, between maybe 1.2 and 1.5% of the genome. So instead of 3 billion base pairs, 30 plus million base pairs maybe. So a very substantial economy of scale. The downside of it is it's a little bit more laborious than if you just directly sequence. Secondly, this capture is not a perfect method. Not all exons necessarily get equally captured, equally represented, so the libraries wind up sometimes being biased. You don't necessarily truly sequence all of the coding regions. And finally, of course, there's no law that says that medically relevant genetic variants have to reside within the protein encoding components. So there are mutations within introns. Presumably there are mutations in regulatory sequences and elsewhere that you would miss. What's happening, I think, right now is this technology was particularly suited to a point where the cost of sequencing was still relatively high, and it was a big deal if you could essentially reduce your sequencing needs by a factor of almost 100. That's becoming less and less of an advantage now. We do most of our clinical sequencing at Hudson Alpha, which I'll say more about later, in Huntsville. They have more or less stopped doing exomes and do only whole genomes. And so you can actually now extract information from an entire genome, although I will say when you do it, interpreting the genome is a challenge, not so much because of the volume of data, though that's true, but also because it's really difficult to sometimes annotate what the significance of particular variants is, and I'll come back to that point in a moment. Now, if you're in the business of trying to identify a gene associated with a particular phenotype, one question is, how do you actually validate that you found the gene you think you have, that you really are making a correct diagnosis? And there's various ways to conceptualize it. One is, and what's depicted here are sporadically affected individuals from 3 families. And realize that if you do a whole genome, you're typically going to encounter something like 4 million genetic variants of that individual compared to the reference sequence. So you have to filter them somehow. You can't digest 4 million variants and scrutinize them each individually. First thing you usually do is go to databases of frequency of variants and discard anything with a frequency greater than, and you can take your pick, say 1%, figuring if you're dealing with a rare disorder, you're not going to be working with a common variant. But having done that, even if you're filtering out the vast majority of 4 million, you could still be left with a few thousand variants. There are computational tools that will make an estimate of whether a particular variant is likely to have a significant effect on the function of the gene. Stop mutations, for example, that cause premature termination of translation have a greater chance of being pathogenic than missense mutations that just change the amino acid, for example. But these are not perfect algorithms, and they often miss things that are pathogenic or cause things to be thought of as pathogenic that are not. So ultimately, you need some criterion for deciding that you actually have found what you're looking for. One way is to compare individuals across different families, and this is a kind of diagram showing that the first individual has lots of variants, the second individual lots, and the third lots, but they overlap with only a relatively few. And I'll show you in a few minutes how we can actually accomplish that, because so often we're dealing with just one family, and where do you find these other families? But that becomes a really important issue. Second thing that can be helpful is sequencing both parents in addition to the child, because if you're dealing with a rare disorder, many of them will turn out to be de novo mutations. Every time a sperm or egg is made, there are about 30 mutations compared to the parent genome. Most of them are not medically relevant, but some of them will be. And if you find a variant that is new in the child, not present in the parent, the child's the only person in the family affected, that comprises significant evidence that this may be relevant to the phenotype, although you need to prove that the parents are truly the parents, by the way, which isn't always the case. Another way is if you have a dominant disorder, like depicted here, does it segregate in the family? In other words, if this phenotype is present in multiple generations and you find a variant in any one person, now you can go back and ask, well, do all affected people in this family share that same variant and unaffected people not, realizing the phenomenon of non-penetrance can fool you sometimes. So a person might carry the variant yet not have developed the phenotype, or maybe not yet have developed it, so you have to factor that in. And finally, you can look at outliers, individuals who have a phenotype versus those that don't, and if you have a good-sized collection of them, do all the people with the phenotype tend to share this variant, and those that are at the other end of the spectrum not. It's turning out it is a kind of tricky business, and American College of Medical Genetics and Genomics and the Association of Molecular Pathologists have created kind of a schema of how you classify variants that range on the left side from pathogenic, meaning there's very convincing evidence that a particular variant is relevant to a particular phenotype. The other end of the spectrum is benign. Lots of variants, probably most actually, have no measurable impact on phenotype, but you'll see them when you sequence somebody. And in between are the tough ones. Likely benign, somewhat self-explanatory. Likely pathogenic means the evidence is not airtight, but it's quite convincing. Occasionally, as more evidence comes in, likely path will move either to the left towards definitively pathogenic, or once in a while to the right, meaning that we tend to be cautious here. And sometimes a pretty convincing case turns out not to hold up. The tough ones are the middle, the so-called variant of uncertain significance or unknown significance. VUSs, they'll often be called. And they're very problematic because this means there's just not enough evidence. You found it in one person, and computationally it looks convincingly pathogenic, but it's never been seen before, and you have no way of proving that it's pathogenic. And we have encountered lots of instances where people who got results that were VUSs were told by somebody that they had the condition that this test looks for incorrectly. And I tend to say to them, you know, what about the term variant of unknown significance is confusing? But the answer is it is. And yeah.
Can I ask you about a variant? If there's a single base pair change, is that defined as a variant, or is it a proportion of change within the gene?
No, so we're talking here, a variant could be a single base pair change, but it could be longer than that. It could be a deletion or duplication. It's a difference between the sequence in that individual compared to a reference sequence.
Sure.
Yeah.
When you get these VUSs, which is a new term to me, is it always in, the same region that you would have expected, say, the pathogenic variant? Or can it be in a different region? Like, is one going to be in an intron and then one in an exon? Or, when you do computationally these studies?
So there's no rules about it. A VUS just means that the variant, wherever it was, is not definitively proven to be pathogenic. And things in introns have a pretty high probability of being VUSs because introns are not conserved to the degree that exons are. And you'll see lots of variation there. Proving a variant in an intron is pathogenic unless it's at the splice donor or acceptor, places where we know that splicing can be altered, that can be really tricky. But you'll find plenty of USs in exons. Most of them are missense variants, things that change the amino acid from one to another, which sometimes has a profound effect and sometimes none at all. Sometimes it actually doesn't affect the coding sequence, but it does affect splicing. There's lots of ways things can cause havoc in the kind of reading of the genetic code. But then again, many of them are going to be completely meaningless. And just looking at one, in fact, I think the next slide shows you, and it's not meant to go through the details, but it gives you an idea of how complicated this can get. So ACMG put together this schema of levels of evidence, and each of these codes corresponds to a kind of evidence. I don't remember specifically what these are, but very strong evidence might mean that this segregates in the family, that there have been multiple individuals found with the same variant who have the phenotype, that it's de novo and both parents have been biologically proven to be the parents. Those kinds of things would constitute really strong evidence. On the other side of the coin, there's evidence of something being benign, which is it's been found with appreciable frequency in the general population without evidence of the phenotype, for example. And there's an algorithm now, which is what's meant to be shown here, all the kinds of evidence that collectively would convince you something is pathogenic, likely path, benign, likely benign, or of uncertain significance. So it's become a pretty formalized process and takes a lot of skill as an analyst to weed through this and make a conclusion. And most of you, I mean, some of you maybe will get into that, but most of you would be more in the line of consumers of this information. And the message is, if somebody says it's pathogenic, there's really strong evidence. Somebody says it's likely path, we will return those data to families, but we'll put a little caveat in there that more evidence needs to be generated. And if it's a variant of unknown significance, we sometimes will return that too, because oftentimes there's pretty convincing evidence, but with a lot of caution, and we would not use it for medical decision making, like, for example, prenatal testing in a future pregnancy, for just one case in point. Question?
Yes. At what point does a phenotype become considered a pathology, or is there any kind of distinction between those two terms, the way that they're used in this context?
Well, see, I don't think that there's an ironclad definition of what constitutes pathology, because it depends on the focus that you're looking at somebody with. I guess it really gets you into the kind of philosophical question of what is disease, and what might be a maladaptive physiological circumstance in one environment might actually be an adaptive one in another. So I'm not sure I know the answer as to how you define disease, but what we're really talking about, and I think the next slide even gets, let me jump ahead here for a second. You're generally doing this with individuals who have some, I guess you could say, maladaptive phenotype, developmental disability, autism spectrum disorder, a particular medical condition, kidney disease or liver disease or whatever it might be. So the phenotype is pathological because there's some sign or symptom that is associated with it that is not viewed as positive by the person. Doesn't mean it's genetically based, obviously. Of course, there'll be many that are not. But it's up to the clinician to decide that something is pathological. What we're asking, and maybe this notion of pathogenic in some ways is a bit misleading, all we're really asking is, does this variant explain this phenotype? It's up to you, the clinician, to decide that the phenotype is a disease, which I guess that's a longer discussion as to what constitutes disease. I just wanted to, since this came up, we use now what's called the human phenotype ontology. It is a organized nomenclature for defining phenotypes. HPO is the term it goes by. There are a variety of ways of accessing it. There's an internet program called Phenomizer. We are beginning to use a sort of commercial system called Phenotypes. There are a lot of ways of getting to it. But it's a way of describing phenotypes with a higher precision than these are all things that involve dysmorphology, but they're not limited to that. So if you were to see, syndactyly, kind of fusion of fingers, there's a term that is recognized now, and that's how we define phenotypes. Let me just go back one. Question could be asked, how do you get to the point of taking an N of 1 and expanding it to a larger number? And, we're dealing very often with obscure disorders. And this is a diagram of what's called matchmaker exchange. It is a variety of different databases. One that's commonly used is GeneMatcher. We're now using, because of our association with the Undiagnosed Disease Network International, something called Phenome Central. But they're fundamentally the same. Actually, MyGene2 is for consumers. You put a phenotype in, defined by those HPO terms I just described, and whatever genetic variant you have found. You actually can upload a whole genome sequence in there, but mostly we don't do that. We find a variant that we think might be pathogenic, but it's never been seen before. And what happens is anybody anywhere in the world who has entered the same phenotype or the same gene or the same variant, could be any of those, in any of these gets matched to you, and you get an email that says, match was found, here's the email address of the person who entered it, and then you can contact them and compare notes and decide if you think this phenotype is similar enough that oftentimes you will expand an N of 1 to an N of many through this. It's turned out to be a remarkably useful thing. We've now had several instances in our program where we found something we weren't really sure was pathogenic, but it looked awfully convincing, and ended up on GeneMatcher and made a match, and lo and behold, it was somebody who could have been a SIP of your patient, but was in another country much of the time. And we've had a couple of now publications resulting from that, and I can tell you, actually, as editor of a journal, a lot of the cases we get are GeneMatcher or matchmaker exchange matches. And it is a HIPAA-compliant system. Our consent form, when we do sequencing, includes data sharing of this kind, and people want this to happen because it's a way for information to ultimately lead to a diagnosis and to convert something from a VUS to pathogenic. Very quickly mention, we do have an undiagnosed diseases program here. All the different staff, core staff, I should say, are shown here. We have a cadre of consultants outside the core staff, both in pediatrics and in adult medicine. We see patients with chronic undiagnosed disorders that usually are people who have been looking for an answer, sometimes for years. They don't always have genetic disorders, but a high proportion of them do. And we try to do multidisciplinary evaluations, collect medical records, and ultimately then create a kind of plan for evaluation. These numbers are a month or two old, but the spirit's about the same. In about 5 years' time, we've had almost, probably by now, have had 500 referrals. We do a review and triage. Many of the people who are referred will refer out to other programs. If it's a pretty clearly neurological problem and they haven't really seen a UAB neurologist, we'll send them there first rather than do a multidisciplinary evaluation. Out of just over 200 evaluated when this table was put together, we had diagnoses in 80 of those. So realize these are people who have already been worked up extensively and unproductively. So it's not a bad diagnosis rate, about 40%, I guess. It's one of the first ones we made. It was a child who had a seizure disorder, hypoplasia of the cerebellum and of the pons, and very profound developmental disability. Had all the genetic testing we could think of, and it didn't give an answer. And then when we did sequencing, you actually found two different de novo variants, one in this gene SPTAN1 and the other in SETD5. The child had a duplication of, I guess, 8 bases in SPTAN1, and then a splicing mutation in SETD5. Both parents, who were proved to be the biological parents, did not have either of these variants, so they were two new mutations. They both have been implicated in pathology. We're pretty sure the SP10-1 was the main driver here. This is a published case with the same mutation, it turned out, also with cerebellar and pontine hypoplasia, maybe not as severe as our patient, but still severe. It's a cytoskeletal protein. And one of the things that now we have the opportunity to do, and which we're trying to work out, is creating a database of these variants, because there may be investigators on campus, in this case, interested in a brain spectrin-like protein, and we can offer a pathogenic variant and look at what the function is, which is still largely unknown. This is a pie chart of the kinds of cases, and there's a bit of bias here. So neuromuscular is a big component of diagnoses. I think that's because one of the early diagnoses we made was a neuromuscular problem, and it caused the neuromuscular clinic to refer lots of patients to us. They said, oh, this is cool, we can actually get answers in some cases. And you know, that has really been very productive. Doesn't mean we can always treat them. Sometimes it's just an answer. We had an instance, just quickly give you an anecdote, a 60-odd-year-old man with weakness, and had been seen for years, both here and had gone all over the country, and no answers, but lots of sort of comorbidities. He had diabetes, he had at one point a statin-induced myopathy, he had spinal stenosis, a lot of reasons for weakness that were dismissed. And the thing that bothered us in terms of just assuming it was any combination of those things was that his CPK, creatine phosphokinase, was just a little bit elevated. And he had had a muscle biopsy and nobody found anything. We didn't sequence him, we did a panel of tests for neuromuscular disease. Turned out he had a myotylin pathogenic variant. And then we went back and asked him to do another muscle biopsy. They stained it appropriately, and sure enough, he had a myopathy that's rare, but fit pretty well with his phenotype. Can't treat it, but he was actually practically in tears when we told him, not mourning the fact that he had this diagnosis, but more the fact that somebody finally validated his concerns and that he had a reason for weakness other than all of these other morbidities. Why is it helpful to establish, and why is it so hard? I'm going to divide this into clinical and personal utility. Clinical utility, short of actual treatment, could be guiding further evaluation. What should you be looking for? What's the prognosis? Then there's personal utility, such as peace of mind, which I think is what that individual developed, and really the idea that it helps you know what you're dealing with, even if it's not something you can treat. Why is it hard? A lot of times we get referrals of people with non-specific symptoms. Care tends to be fragmented. Sometimes it's the obscure presentation of a known disorder. Other times it's a vanishingly rare disorder. I guarantee that SP10 variant that I described would not have been diagnosed without sequencing. No way that we would have thought of that. Many people lack access. It takes time. This is a very labor-intensive process. New tools, sequencing, for example, are allowing diagnoses to be made that would never have been made before. We see a lot of people that have what are probably psychosomatic complaints that we try not to do those analyses, but it's sometimes hard to be completely sure. Some things we've learned, sometimes the diagnoses you make by sequencing are obvious and you kind of kick yourself and say, why didn't I think of that? And the only comfort is usually at the end of a long line of people that didn't think of it. And it's probably true, some of these are obscure things. If the right person who had just the right experience has seen them, but how do you assure that that's going to happen? Others, like I said a moment ago, would definitely never have been diagnosed in the pre-sequencing era. One thing we've learned is the phenotyping is at least as useful after the sequencing as before, meaning that the phenotyping is never as complete as you wish. And then when you find a variant that you're trying to validate, you go back and say, we expect to see the following in this with this variant. Do we actually see it? And if we do, then that helps to validate that it's relevant. So sometimes the phenotyping more precise after the fact, and we certainly don't always get answers. Is it possible for somebody to pull the shade on that right-hand window? There's a reflection coming from a car, which is sort of proving to be dazzling. Thank you. Just want to mention, we have a project that we do with Hudson Alpha. We call it South-Seq. It's part of a NHGRI-funded CSER project. CSER is an acronym for Clinical Sequencing Evidence Generating Research. It's essentially designed to provide the evidence to validate genome sequencing. We're sequencing sick babies in the special care nursery, trying to establish diagnoses. We have about a 30% diagnosis rate, it's turning out. And this involves a clinical trial where we're randomizing babies. Half of them get return of results, or their parents do, through a genetic counselor, which is the traditional way, and half through nursery staff that we train. And it's meant to ask, can we scale the skill set necessary to a larger group? There are now therapies too, and so it's not always just telling people the name of what they have. Tuberous sclerosis complex and Mendelian dominant. Turns out the genes involved, when either of them are mutated, lead to hyperactivity of mTOR. Everolimus is an mTOR inhibitor, and we can shrink the subependymal giant cell tumors or the renal angiomyolipomas now with a drug. I don't send these patients to surgery anymore as I used to. Or in cystic fibrosis, a chloride channel, there are so-called correctors that prevent degradation in the proteasome, or potentiators that enhance chloride channel function in the presence of mutation. The first FDA-approved drug, ivacaftor, you can see a dramatic improvement in pulmonary function if you have this particular mutation. And what the community is now doing is whittling away mutation by mutation, finding drugs, either potentiators or correctors, that will achieve this similar kind of outcome. Now let me turn attention to common disease for a moment, because the monogenic disorders, those are the things we've talked about so far. Common disorders, things like asthma and hypertension and so forth, are much more complex in terms of their genetics, because what genetic components there are usually are multiple genes interacting with each other in the environment. And the hope here is that if genetic liability is recognized early in life, that you can either predict who's at risk and then prevent them from climbing this curve, maybe with influence of the environment, into the disease category. So the question is, can you figure out from the genetic liability who's at risk and offer them some plan to modify that risk? And for a long time, the mainstay has been association studies looking at single nucleotide variants here, and this is a C instead of an A at this particular allele, and you do case control studies. A hypothetical one is shown here, where you can show that the allele 2 in this case is more common in a group of people with, say, asthma than not, and you can calculate from this an odds ratio of the chance of getting asthma given the presence of allele 2. This was the first example of a genome-wide association study done showing complement factor H being associated, I guess it was over here, with age-related macular degeneration. No one had any idea that that would end up being related to that phenotype until this association study showed an association of a variant in this gene with that phenotype. And over the years, each of these colors is a different disorder, where GWAS has shown the association of a particular variant with, or sometimes multiple variants with a particular disorder. So there's a very dense catalog now, but the actual risk associated with the presence of a variant is generally very modest. So if you consider a single variant, and this is all hypothetical, the red is meant to be the variant that's associated with disease, and you compare the risk of disease in a person who, say, has one copy of this variant to somebody who doesn't, what you'll see maybe is an exceedingly subtle, statistically valid maybe, but subtle change in risk in the presence of this variant compared to when it's not there. But the actual odds ratio of disease could be 1.1, 1.2, and you're dealing with a common disease, and is it going to really matter to somebody to tell them that they have a odds ratio of 1.2 of developing, say, coronary artery disease? And the answer is, most people would say probably not, but those are just two small increments of risk. So what's emerged in the last well, at least kind of visibly in the last couple of years. I think it's much longer in the making, but has really become sort of mainstream, are what are called polygenic risk scores. And what's turning out is that if you have, in this case, hypothetically 4 variants depicted by the different colors here, each of these operates independently of the other. And the risk associated with any one is additive to the risk associated with any other. So if you compare somebody with 4 variants to none, it actually causes the change in risk to be quite meaningful. In fact, sometimes in the same range as you would see for a single gene Mendelian disorder. So these polygenic risk scores are very quickly moving from kind of theory to practice. And I'll tell you later, we're going to launch a pilot here because they have the opportunity to identify individuals at risk of coronary artery disease, type 2 diabetes, obesity, many other things that are much more powerful than any other tool so far that has existed. Of course, it raises the question, can you modify the risk? And the answer is sometimes yes and sometimes not yet. Yeah.
In the case of the polygenic risk panels, right, how have these alleles been identified? Have they all been identified individually, and then it's just somebody putting them together into a panel?
Well, basically, I mean, they are individually identified. You need to show that they're not in linkage disequilibrium with each other, indicating just a chance association and that they would not operate independently. So you need to demonstrate independence. The key thing you have to demonstrate, actually, is that they're relevant in different populations, or if not, that if, for example, it's been found, as probably most have been in a European population, that you don't apply it then to a different population where you're going to probably not find the same association. And it's raised the issue, a very critical one, that many of the studies done so far have really not been in diverse populations. And as a consequence, we have a very biased understanding at the moment of how these are to be used. So that's been a real concern. Let me move quickly through a couple of other things and then get to some projects going on here. You're probably aware that there's been a kind of revolution in genomic analysis of cancer. Cancer is a genetic disorder. It's the accumulation of genetic change which gradually makes the cells more and more abnormal in their behavior, which comprises ultimately what leads to a metastasis. And one of the tools that has become now pretty mainstream is doing genome sequencing of the tumor, ideally compared to the normal DNA, although now we have enough knowledge of some of the tumor-specific variants that you don't necessarily need to compare it to that person's non-tumor DNA. And it gives you a catalog of tumor-specific genetic changes, some of which are known to be major drivers, and then the possibility of matching therapeutics to the particular genetic change. Here too, the issue is that it's a lot easier to catalog what's there than it is to find drugs that necessarily are successful at treating these things. The other thing that's become interesting is the ability to do so-called liquid biopsy. As cancer cells grow, a fair number of them die and release DNA into the circulation. And that DNA then is derived from the tumor, and you can actually now do sequencing of DNA in the circulation. And by the way, we all have some DNA, free-floating DNA in our blood, which is derived from cells that naturally die and release that DNA. You may know that in pregnancy, this has become a way of doing prenatal diagnosis because fetal tissue will release DNA into the mother's bloodstream. Well, tumor DNA does that too. And you can sequence the DNA found in circulating blood and sometimes identify a rearrangement associated with cancer, either before the cancer becomes clinically evident or if it is already known, using it as a marker of progression and response to treatment without having to biopsy the actual tumor. We digress for just a few minutes and make a few comments about epigenetics. You hear a lot about it. We help you to kind of know what we're talking about. The term was coined by C.H. Waddington, a developmental biologist, I think in the 1940s. And it was described as a heritable change in gene expression that it's not associated with a change in DNA base sequence. Now, heritable here doesn't mean from person, parent to child. It means from one cell division to another. That's an important point to realize. And at least at the molecular level, the signature of this tends to be the occurrence of methylation of DNA, and especially the occurrence of 5-methylcytosine, which is the methylated version of the DNA-based cytosine. And when that happens, which is depicted by these green things in a promoter region, it tends to silence the gene. When it happens elsewhere, it can have other effects, but the promoter methylation, and there are these so-called CPG islands, that is cytosine followed by guanine in promoter regions. And it's a way for the cell to more or less permanently turn off a gene. If it methylates the promoter, that methylation is propagated. And the way it's propagated is, here 5-methylcytosine is depicted in red, and the guanine that follows it in yellow. That would be normal, this would be the base sequence. And of course, this reads the same in both directions, right? Because C and G are going to base pair. When DNA replicates, there's no pool of 5-methylcytosine. And besides, how would the cell know where to put it? Because it's going to pair with guanine anyway. So what happens actually is when the DNA replicates, the blue are normal cytosines that now bind to the guanines in the two daughter strands. But there's an enzyme system that recognizes what you could call hemimethylated DNA, because one strand from the parent still has the 5-methylcytosine. And the enzyme sees that, and what it does is, to the next door cytosines, it adds a methyl group and methylates the DNA. And so where methylation has occurred, that methylation is propagated. And there's a lot of complexity to this. The histones, which are the proteins that bind to the DNA, are also methylated, and other modifications, all of which basically controls whether chromatin is in a closed, inaccessible state for transcription or in an open state. And there are various proteins, I guess they're shown here, complexes that recognize the methylated or unmethylated DNA and block transcription to the methylated DNA. So largely epigenetics amounts to usually methylation-based changes in the accessibility of particular genes to transcription. And it's become of interest because environmental factors can cause particular loci in particular tissues to be methylated and then permanently turned off. And that can have an influence on future health. There's, for example, evidence that during in utero life, if a fetus is exposed to a condition of starvation where it becomes disadvantageous to store sugar in the form of glycogen or fat, and therefore insulin tends to be suppressed, that suppression can be a lifelong phenomenon due to methylation of genes involved in control of insulin production, and the long-term effects would be diabetes and metabolic syndrome. Realize that there is no single epigenome. Each tissue has its own epigenome because the genes turned on and off in your pancreas will be different from your brain. So you could do an epigenetic analysis of blood, but it only tells you about blood. Quick word about pharmacogenetics. If you conceptualize the process of absorption, metabolism, and excretion of a drug, many of those events are controlled by various genes. There are phase 1 reactions where usually the cytochrome P450 system is involved in oxygen related reactions that may either activate or inactivate certain drugs. And phase 2 reactions are conjugation reactions that affect solubility. There are a variety of genes that have been associated then with either changes in the ability to activate or inactivate drugs or to excrete them. We tend to think in terms of what a term that might not be familiar to you called diplotypes. So if you consider two genes, A and B, that are near each other on the chromosome, or two loci within a gene even, and A1 and B1 are on the same chromosome, that's called a haplotype, A2 and B2 as well. The genotype would be heterozygous A1, A2, or heterozygous B1, B2. The collective set of haplotypes is called the diplotype, and you can have specific diplotypes of particular variants in particular genes that are known to be associated with specific ways of metabolizing drug. The various cytochrome oxidases, for example, especially CYP2D6, are associated with changes in metabolism of multiple drugs. And it explains why a standard dose of drug might be toxic to one person and ineffective in another. And the idea would be, well, you can predict how a person will respond by doing their genotyping. Warfarin is metabolized in part by CYP2C9, and also there's a polymorphism in its target, VCOR-C1, which affects the degree to which the warfarin actually is effective. And you can predict an individual's responsiveness to warfarin then by genotyping this VCOR-C1 and CYP2C9, or clopidogrel, antiplatelet drug, is really inactive until CYP2C19 activates it. And if you have a variant in this, it will cause you not to activate it, and you won't respond to the drug. There's some HLA variants associated with toxic epidermal necrolysis or Stevens-Johnson syndrome. There are places in Asia where it's so common, everybody gets genotyped for this HLA variant, and they carry a card. And if they find their way to the emergency room, say having seizures, the people there will know not to treat them with carbamazepine. And it's plummeted the frequency of that complication. Not so easy in other populations because no single HLA genotype seems to be associated outside of Southeast Asians. There's a pharmacogenetics knowledge base online where you can find information about any particular variant. And there's a clinical pharmacogenetics implementation consortium, CPIC, which rates different variants as to their clinical utility, which is worth knowing about. So in the last 5 minutes, I want to tell you about two programs that we're involved in here. One called the Alabama Genomic Health Initiative, which is funded by the state. We've now received $6 million over the last 3 years. We're hoping it'll be $10 million over 5. And there's various reasons we're doing it. We think it is serving as a way to engage people in the state in genomics, both participants and health providers. And as you'll see, there's a research database. We recruit 2 cohorts. The population cohort are just adult volunteers not selected for any particular phenotype. The affected cohort are people with phenotypes. The former have genotyping using a microarray. For under $100, we can genotype about 700,000 single nucleotide variants. The affected cohort get whole genome sequencing. Many of these are the patients we see in the Undiagnosed Diseases program. People are given a chance to opt into having their DNA stored in a tissue bank, DNA and blood actually, included in a genomic database. And if they're UAB patients, their medical records get linked through the I2B2 system. We have a variety of working groups. I won't have time to go through what they all do, but it's a very active project where we have frequent meetings of these groups and discuss a lot of issues, bioethical issues. We've created an education program. We are looking at the specific variants we find and trying to adjudicate their pathogenicity. We have tables you may have seen set up in the Kirkland Clinic and a few other places people can enroll. They get consented. It's an IRB-approved protocol. Testing is done at Hudson Alpha, and then they get a return of results. We have been working really hard to increase the diversity of the sample, opened up pop-up clinics around the state. This is now 2 months old. I know we've exceeded 5,000 participants now. I hope this one county that we hadn't filled is going to be filled in because they've just opened a pop-up. It's Monroe County, and they've opened a pop-up clinic there. So this week that should be, we should have every county. And we return results of actionable variants. These are a set of genes that if you have a pathogenic or likely path variant, It predicts disease with high probability, and there's something you can do about it. Many of them are cancer predisposition syndromes, connective tissue disorders that predispose to aortic dissection, various cardiac disorders, a few metabolic conditions, and malignant hyperthermia. The ones in red, we've returned results. That gets done through a genetic counselor. Our rate of return is about 1.4%, which is right where we thought it would be. We had estimated between 1 and 3%. There's also a family history questionnaire. Our genetic counselors review it. And some people who have non-informative findings will still have a family history that's concerning, and we notify them about that. There are actually, as of when this was done, which is now 2 months old, over 3,000 DNA or blood samples in our data bank. 92% of people consent to having their samples stored. We, at that point, had 300 and some odd whole genome participants. It's over 400 now. And at that point, we had returned 49 primary variants. This is a list of some of the diagnoses made. I won't have time to go into detail. And right now, we're working on an outcomes assessment. We've been surveying people to find out how useful this was to them. We've developed a way of using the data to help people kind of get a glimpse of their continental ancestry. Some people are interested in that. We're talking about pharmacogenomic return of results. We've set up a portal, which should be launched shortly, for doing these association studies. We have a database now of regional allele frequencies, and we are now about to launch a polygenic risk score. This is an example of coronary artery disease, where high scores give you a very high probability of disease. And so our plan is to create a pilot to do that. And then finally, just say that we are very actively involved in the All of Us research program. This is part of the National Precision Medicine Initiative. The goal is to enroll 1 million people around the country. They share their electronic health records. They get some basic biometrics collected, height, weight, and blood pressure and stuff. They give a blood and urine sample and then fill out questionnaires. Some of them will wear fitness trackers. It all gets sent to a central database. The data center is at Vanderbilt and a company called Verily and the Broad Institute. The biobank is at Mayo Clinic. There are various other components to this, including everybody eventually will get their genome sequenced, and they will have access to all the data, including the raw data if they want it. There's a lot of moving parts. We are here. We're probably the biggest of the health provider organization networks. It includes all the CCTS partners actually in this, and you've probably seen tables maybe in Kirkland Clinic and elsewhere where enrollment is going on. Enrollment is done online at joinallofus.org. There is a participant portal where you can see where you stand in relation to other people in terms of how you answer particular questions. And there's a researcher workbench in process from Vanderbilt that will allow you access to the data to test a hypothesis that's of interest to you. Lastly, let me say we have a Center for Genomic Medicine collaborative with Hudson Alpha in Huntsville, 100 or so miles north of here, really world-class genomics facility. Together we have a T32 in genomic medicine. It's a postdoctoral training program, 2-year training program that involves research, participation in clinics and clinical lab events, seminar series. And this year, we're just launching an R25 summer program for 12 students, undergraduates from historically Black colleges and universities who will spend this summer at Hudson Alpha, next summer either here or there, doing mentored research. And this is ultimately it'll be a 5-year program. And I'll end, I think, only 1 minute late with this now. Probably well-known quote, but one I think is really apt here, which is this notion that who knows exactly how all this will take shape, but Alan Kay, by the way, was essentially considered, I don't know about the inventor, but the father of the laptop computer, which is a remarkable thing in itself. The best way to predict the future is to invent it. So that's what you all get to do. All right, thanks. probably don't have time for questions, or do I?
At least enough time for me to get my presentation up.
Go for it.
I
Okay.
got a quick question.
Yeah.
So clinicians, so we order some of these tests, and I've seen on occasion when we get a variant that's described as pathogenic, it will give us data like this collection of SNPs is seen in X% of your population, X% of the general population. If that information is not included, then can we assume that it's relatively high, the variant that is described as pathogenic?
So it would probably be necessary to see the exact context you're talking about. If they say it's pathogenic, and if they're using this kind of schema that I talked about, then there's a lot of things that went into that, and they're probably quoting some of the evidence that was included in that. Among those will be population frequency, and there are databases now of the frequency of variants in different populations, and they might say that this is less than 1% in exact or in Nomad, other databases that are commonly used. By itself, I think that has to be interpreted cautiously, because you might think, for example, that say a variant that's 0.02% in the exact consortium database, that doesn't mean it's not pathogenic, right? Because sometimes, first of all, if it's a recessive, it only is pathogenic when it's homozygous. You expect it to be seen in some unaffected people. And secondly, incomplete penetrance can occur. And thirdly, for many of the phenotypes you may be interested in, who knows how many people in the exact database have that phenotype, and it wasn't reported because that wasn't why they were sequenced. So, a lot of variants are going to be found occasionally. Cancer predisposition would be a good example. If you find a cancer predisposition gene and they say it's affecting, 0.2% of the population, it could very well be pathogenic, because other people probably are at risk of cancer and don't know it.
Can you just say a few words about the computing power? You mentioned that early on. It seems like that would be a limitation if it wasn't advanced enough to incorporate all the data that you generate.
Yeah, well, it does take a pretty concentrated effort in terms of computational analysis, and so some of it is just raw computing power, because you are grinding a lot of data, and every once in a while we'll get an email from Hudson Alpha that says the system crashed, or somebody was trying to load too many genomes at once. So it isn't, it's not foolproof, and can have glitches occasionally, because the amount of data we're talking about is really massive. That being said, I think it mostly works pretty well. And we do have access to computing. Maybe you'll hear more about it in the next hour, but we do have access to high performance computing, and there are various programs, commercial and in-house developed, that allow you to do whole genome analysis. So it's not easy in the sense, I think when we first started doing this a few years back, the initial way it was done was we got these spreadsheets, and we were doing exomes, so there might have been, 500,000 variants. So those are the biggest spreadsheets I'd ever seen, and they were, think of 500,000 rows, and trying to find something that you cared about was really hard. There are now filtering pipelines, they call them, some of which you can buy. And for a while we bought one. Now we work with Hudson Alpha, and they have a homegrown one that really make this a much easier process. And I think if you do analysis using those systems, what you find is sometimes the variant that's pathogenic just immediately hits you over the head, and it becomes obvious. In a half hour, you scan it and you have your answer. Other times you can go for hours, sifting through variants, and you see something no one's ever seen before, and how do you really know? And you have this nagging feeling, if you eliminate it, maybe you've eliminated the answer, but you can't chase down every single one. We had a recent case where it was a mother-daughter with a phenotype that also affected other family members. It was obviously genetic, and they didn't find anything. And I said, go back, because you've got to find something. And they did. And so there are plenty of examples where the first round of analysis didn't give an answer, but then when you went back, sometimes just looked harder, sometimes with time passing and more information, including better annotation of variants, something new. I don't want to take time away from that, so leave it at that. All right, thanks.
Is it typical to take a couple of minutes break between?
Yeah.
Well, yeah, if anyone needs by all means, let's take a couple of minutes. My watch is from the 50s, so a minute and a half worth of variance is completely within normal limits. For those online, I'll do 30 more seconds to let people settle in the room, and then we'll start. Okay, so my name is Matt Wyatt. I'm the Director of Clinical Research Informatics for the UAB Informatics Institute, the CCTS, and the Health Systems Enterprise Data Warehouse. I am not Jim Semino. He is the person who would typically give this presentation, but he is traveling. I just imagine, if you will, a very highly regarded and highly ranked physician who is a fellow and member and founding member and editor of all kinds of stuff, and was literally working with the people who invented this discipline. Brown, Harvard trained, Mass General, Columbia, NIH, and now UAB. So I'll apologize, I'm not him. I've kept his slide deck more or less intact. Removed a couple of things that I really had no idea what he was talking about. So it wouldn't have been productive anyway. So, but I did, in all honesty, leave a good bit of his content in here for reference. And it is also a matter of the perspective of when we're talking about what is informatics. It is a very broad topic. It really depends on who you ask. There are active discussions in various informatics communities as to what is informatics. And the bulk of this is going to focus on the sort of broad picture that is sometimes described as biomedical informatics, but largely on clinical and health informatics. And because my title says clinical research informatics, that's what I do, a good bit of sort of the clinical research space. I will touch on a lot of things that are contiguous to what Dr. Korf talked about, but most of the conversation is not going to be about bioinformatics. There's a whole domain in bioinformatics, computational biology, molecular and genetics oriented informatics that is tightly integrated with almost everything that Dr. Korf was talking about. hopefully you are interested or willing to listen to things in the rest of the informatics spectrum, because I'll talk mostly about that. So has anybody seen this sort of diagram before? It is a old Chinese proverb that the blind men seeing different parts of a single entity and perceiving it as very different things. If you are touching the tail of an elephant, it feels like a rope. If you are touching the tusk, it feels like a spear. And informatics is very much like that. So it really depends on what your interests are, what part of informatics touches your discipline. It could be anywhere from hardcore computer science and programming, if you're doing computation on analysis pipelines. It can be very much user-centered things within clinical practice. There are areas of informatics in nursing, imaging, pharmacy, pathology, computational biology, a natural language processing, machine learning, text mining, software development, user experience, visualization, analytics. There's a whole host of informatics in the spectrum of everything from basic science to clinical research to population health, and everything in between. So the key point is that while I may talk about 2 or 3 of these things more than others, informatics is a pretty broad discipline. So by all means, ask questions that is relevant to your, what parts of informatics touch your discipline. So the core of this presentation really is informatics as a fundamental domain of knowledge representation and knowledge understanding, concept understanding. And the reason this is important is because healthcare, medicine, biomedicine, and research is transitioning from small scales of data, small numbers, and things that can be analyzed visually with an individual person. And it is, and my typical example is the electronic health record was on paper. It is very organized towards a human being reading something. It makes a lot of sense to a human being reading something. But in a world where it's not one physician with one bag going to a person's house and knowing everything that has ever happened to a patient, to a whole cadre of people caring for a patient, and not just one patient, but a whole host of patients, and wanting to deliver information at the right time, at the right place, in a world where it is impossible for one person to house the entire domain of knowledge necessary to practice medicine in their head. It is all about delivering the right information at the right time. It's all about representing the right content from that interaction so that it can be used by other people downstream, and so that it can be used to promote research and innovation. So when we talk about informatics in this core context, it is really about how you represent information, how you understand information so that you can use the information effectively in a way that is more efficient than letting one person read one record at one point in time. So we used the example culinary informatics. I'm not sure where Dr. Cimino came up with this example, but it works, and I understand it because I can understand what fruit salad is. So we'll go with that. So which of these is not a fruit? I'm going to need some response from the audience.
Almond.
An almond is not a fruit. So which ones of these would not go in a fruit salad? A tomato, clearly a fruit, but would not go in fruit salad. So it turns out what's more important for a fruit salad is not whether something is a fruit, but its flavor profile. So when you start representing this very simple information, you start to create relationships between concepts, between objects and attributes of those objects. So a watermelon is a berry, it is sweet. So if you get down to tomato, tomato is a berry, but it's not sweet, it is savory. An almond, while it is not a fruit, it is sweet. So, and then you transition from individual objects like strawberries, almonds, and tomatoes to the concepts that represent those berries, sweet, savory, and then the concept of a fruit salad. So you begin to understand the individual pieces of information and what they really mean so that you can put them together. And while a person can read and just logically know that, yes, a tomato is a fruit, but you don't put that in a fruit salad, when you start trying to decide, absent the human being reading that, what to deliver to a person in the context of research or clinical practice, you have to understand what those concepts are, what those relationships are, and a more discrete meaning so that you can build systems, tools, processes in order to deliver the right information at the right time. So this is information that was in the slides previously, and this is just random. These are numbers of Google search results from last year, and from some point in the past. I don't know if Jim had updated his slides last year. Could be the year before. So bioinformatics was already gaining prominence. The concept of bioinformatics was tightly associated with the Human Genome Research Initiative, the development in genomic sequencing and personalized medicine, and bioinformatics was pretty prominent there. I did this again yesterday, and note that the bioinformatics has still increased by at least, it has at least tripled, but the concepts of health, medical, clinical informatics have also increased, and I think this is a product of the explosion in electronic health records, the extreme increase in the amount of clinical data, and the use of that clinical data in identifying not just genetic predisposition, but the rest of the attributes of a person, their environment, their care, and the idea that we want to provide health care and not just addressing maladies one person at a time, one instance at a time. I would point out that culinary informatics has just exploded. They went from 144 to 1.5 million results. That's just crazy. So here's a quick example. Nurse reported that the patient's glucose was 50. The physician ordered some glucose and then repeated the glucose. So the term glucose is in there 3 times. So what does the first glucose refer to? Physiological parameter, medication, or a test? It is a physiological parameter. But the other 2 are clearly not that. One is a medication and one is a test. Same word in one sentence means 3 different things. So you take those glucose and expand those into more meaningful representations of a concept, at least verbally, so that you can begin to understand them distinctly from one another and how they are related to one another. The context, and then the control terminology. So switching from glucose to serum glucose level, intravenous glucose, medication formulation, the test. And then just like fruit and fruit salad and flavors, you can represent a serum glucose level as a physiological parameter. The location is a bloodstream. The associated chemical is glucose. And you can follow each one of those paths. The bloodstream is a very specific thing that has very specific relationships with other things. Glucose is a very specific thing. All chemicals can be associated with very particular entities in medicine. And you begin to have a more discrete, descriptive understanding of those concepts. And the reason that is important is because it is perfectly reasonable for a human being to read that the glucose was low, so you gave some glucose and then you ordered another glucose. But if you want to take action as a system, as a dumb computer who will do whatever you say very fast, but you have to tell it what to do, you have to have that next level of understanding, and that's what this is. And then once you do that for a descriptive understanding of a statement about reality, you can begin to use those same understandings, that same codification of concepts and relationships to then prescribe activity or to guide activity. I should rephrase that. Not to prescribe activity to a clinician, but to define processes and tools and systems for the delivery of information at the right time, for representation of information to the clinician at the right point in clinical operations. So you can begin to create these logical processes that can be valuable, more valuable than a piece of paper where one person can read. That a physiological parameter is low means you do some action. Those actions could be a medication to address a chemical. It could be a test to measure that chemical. I'm really not sure what the transition was in this, but what is the location of diabetes? Is it the bloodstream? Is it the isolate cell? So I can say here, since I'm not a clinician, I literally don't know. I know what 3 of those things are, maybe another one, but I don't even know what all those things are. So I certainly don't know if that's the location of diabetes mellitus. And that's one of the places where there is difficulty when you're starting to talk about disease, when you're starting to talk about disruptions in individual activities and physiological processes in a human body, and especially when you're talking about multifactorial cause and identification of pathway interactions. So how do you accurately represent that if your foundational understanding is that you have a location of a difference or deviation from the norm? So it gets complicated. So the distinction between informatics, biomedical informatics has typically been the highest level sort of umbrella category. Anything, and some people will argue with this, but biomedical informatics tends to encompass the entire range. Anything that in biology or particularly biomedicine, biomedical informatics. So originally it was coined as computers in medicine. That was the original professional organization, was actually called Computers in Medicine. Some of the founders of this particular discipline, Bob Greenis, Ted Shortliff, it was literally the act of applying computers and technology in support of processes in healthcare. So Jim's sort of shortened version, the art and science of organizing knowledge of human health and disease and making it useful for problem solving. And that can be problem solving in research and novel approaches to disease and new discovery. It could be useful in clinical practice. And this can range from bench science, basic science, molecular biology, computational biology, genetics, through physiological cellular processes, through pathways in human and animal models, all the way to first in human interactions, interactions in research. My area is clinical research informatics, so the actual facilitating of research and discovery in human-oriented research, all the way through patient care and clinical informatics, and then on to population health, population science. There's a whole domain of public health informatics that is very much interested in surveillance and predictive modeling and recognition of population level interactions, population level events that require interaction and how to do that. So the spectrum of problem solving for informatics really could be just about anything you want. It's very biology and basic science oriented all the way to population health oriented. But it really is about the collection, understanding, and connection of information and the delivery of that information. So interpreting signals from medicine, whether that's a person speaking, a physiological monitor, a lab test, etc. So the challenge in healthcare is that it is not like banking and finance where there are a discrete number of types of interactions. There's not a discrete type of even methodology for interactions. There are some things that are purely text, some things that are visual, some things that are graphical representation of signals. There's text that exists that is anywhere between ridiculously poorly formed and meaningless, all the way to beautifully formed narratives about the patient that provide the perfect summary and the perfect set of information for the next clinician. Now, my typical joke there is that is absolutely wonderful until 2 visits down the road and nobody ever looks at that perfect note again. But regardless, in the model that we want to make information available not just to one person at a time, taking information from that text and then using it for subsequent actions afterward is extremely important. And in my world, where people want data for research, they don't want me to send them 100,000 documents. They want me to send them particular clinical variables. If those particular clinical variables are buried within free text, that's a real challenge. It is not a solved problem. It is a whole domain of informatics and machine learning, text mining, natural language processing that attempts to solve that problem, but it is a challenge. I'm really not going to read this for you, but how does informatics help? So it's really around, in my mind, the crux of this issue is that there's a lot of data. There are a lot of complex processes. There are a lot of people who are trying to take actions, whether that is research or clinical practice. And really what the challenge is to provide the information necessary in the right context, at the right time, to the right person, and to obtain information from that clinical process in a way that is as unobtrusive as possible. And you can do that in a variety of ways. And it could be expert systems that provide advice or decision support to clinicians, taking into account the exact patient that you are looking at, their relationship or likeness to other patients they've seen previously, their conditions, their physiological state, and how that relates to the world of research knowledge that has been published, and deliver the right information to them at the right time. And then it could be taking all the information that is collected in clinical practice and giving that to the research community in a way that they can make new discoveries. That's the part that I like. So transitioning, really focus on the electronic health record. So the electronic health record is not a research tool necessarily, but it does offer the opportunity for lots and lots of value from a research perspective. So informatics is involved in both the building of electronic health record systems so they work better, representing the content to clinicians so that it works better, collecting all the right information and associating it together so that it works better. And the value in that is that the volume of data in clinical practice is just increasing exponentially. The other value is that as we move forward and do prospective clinical trials, randomized control trials in small populations that are largely oriented around people who look like me, who almost purposefully don't have any quote-unquote comorbidities. The value in that clinical data in very, very broad populations is that you can try to replicate those findings. It is a huge post-market surveillance in the world of medications, but it is a huge validation opportunity for really anything that has progressed to frontline clinical research. So I'm going to switch gears a little bit and talk really about providing people data from the electronic health record. And I'll go through this really quickly, but I want to tie this to some of the things that Dr. Korf mentioned. So if you don't take anything else from this section, take this. We have lots of data. We can give it to you. Come talk to me. Data exists in the clinical record. You're probably not using it as well as you should. You can. Come talk to me. So the models for access. We do, in fact, my area of research is clinical research informatics. So it's a variety of services, people who provide you data with knowledge and understanding of clinical systems and how to provide that data for research. But it's also about building self-service tools. So we have built tools, and I'll show you one called I2B2 that lets you get data yourself. All day, every day, anybody with a Blazer ID can do it. And you can identify populations of patients based on a whole host of clinical characteristics. There are, I think at the last count, 600,000 plus unique clinical variables included in I2B2, and that doesn't include the omics. So you can ask effectively any type of question on 600,000 different types of pieces of information in clinical practice and find out how many patients meet certain criteria. After that, you can download data on those patients. You can get every lab test, every diagnosis, every visit, you can get all the demographics. You can get hundreds of thousands of pieces of information, detailed data, individual lab tests, individual medications, etc. And you can use that at any point in the research process you want. Preparatory to research, feasibility, preliminary data for a grant. You don't have to go to the IRB, etc. With that said, the next step is potentially getting identifiable information. It is recruiting patients. It is going and getting an IRB approval and getting detailed data so that you can either do more research or recruit individual patients, and we can help with that too. We have a whole host of people who are specifically oriented towards research data delivery. And then when it is still necessary to do manual chart review, when it takes a knowledgeable clinician or a knowledgeable person reading an entire chart or portions of a chart to make a clinical assessment, we can facilitate that too. I just don't want people to do that unless it is necessary. So we have a couple of different accesses for via an analyst with an IRB protocol or without an IRB protocol, we just won't give you PHI, or self-service, you can access that data that is effectively coming from the Cerner Millennium Impact System, though it does include information that is not included in Cerner. What's there? Don't look at those numbers. As you can see, I haven't updated it in 2017 because it's really not necessary. It's more than that now. We're well over 1 billion. We may be up to 2 billion pieces of information at I2B2 now. It's lots and lots and lots of data. It includes anything that is in impact, anything that is inpatient, outpatient, any of the ancillary data within UAB. It's not UAB affiliates. So sorry for the folks out there in the CCTS partner network. It is not our CCTS partners. It's not iFoundation Children's. Those are separate legal entities. It is not the VA, etc. Perhaps it can be someday if folks want to work on policy. I'm happy to follow along. So this is just a somewhat outdated example of what content is in there. The basics are demographics, visits, labs, meds, diagnoses, procedures. We have things like the allergies, consult orders, imaging orders. We've incorporated some things related to geographic information systems like the census information, so you can identify patients who live in a census tract with a particular characteristic. You can't actually get the census tract because that is too granular and constitutes protected health information. Various vital signs, height, weight, BMI, blood pressures, pain scores, all kinds of stuff. And if you want something in I2B2 that exists in structured data in the clinical record and it's not there, tell me and we'll put it in.
Quick question. Oftentimes, send out lab tests are in the chart as a PDF. Do those get pulled as well, or not so much?
Only things that are structured as a structured lab result in center. So reference labs that actually end up as a value that is treated like a lab value, they show up. Things that are documents from external references don't. Now, that doesn't mean we can't provide you that information. Excuse me. It's just not structured clinical data that we incorporate. So I2B2 is a self-service system where basically you can find concepts, create queries. This particular one says someone who has that particular diagnosis and has been on a protein pump inhibitor, and I think this query is probably about 3-4 years old now, but it's a perfectly reasonable example. Patients with this and this, you get a number. You can do things based on Boolean logic, value constraints, less than or equal to, greater than or equal to, in numeric ranges if it's numeric. And one thing I want to point out is that this is possible because there is structured data and because it is not in sort of unstructured, human readable, but not machine readable content. The more that is actually in a structured format that is encoded in a way that is understandable, the more we can do in systems like this. And that doesn't necessarily mean, I mean, you can see that there are places where a drop-down list of 5, 10, 15 different pieces of text is perfectly fine. You can actually do searches on text in some places. But the more you have structured data, the more you can actually make use of it afterwards. So that is my plea to everybody. Go to your colleagues and say, why are we putting this in text? If we want to use this again someday, we should put this in a structured place. And the people in the health system will allow you to do that. All you have to do is ask. So in I2B2, you can get aggregate information like this. You can get, you can actually download detailed data like this. So this is another point where this representation of content is very much tied to the fact that we have systems of encoding content and mapping data together that is meaningful. So it allows you to get this content back for your research purposes. And this is, like I said, this doesn't require IRB approval. You can log into I2B2 and pull this level data all day long. You just won't know the actual medical record number and the actual encounter number. But you can know lots and lots of information about the patient population of interest. Oh, any questions before I move on? No? So big caveats. And I often find myself in a place where I'm acting both as the advocate of clinical data as well as the describing the problems with clinical data. And the way I usually try to frame that is that while there may be things that are inaccurate, so there's a potassium of 122. Any clinician in the room knows that no human being ever had a potassium of 122. It didn't happen, can't happen, it's wrong. There you go. It's in the legal clinical record for a patient. It's wrong. So there are things like that. There are pancake people who are 100 pounds but only 8 inches tall, and there are string bean people who are 15 feet tall, and there are things that are wrong. But most of the time when we're talking about accuracy of the clinical record or completeness of the clinical record, how I try to describe it is, it's fit for use, its purpose. It was not collected as a prospective, specifically prescribed data collection for a research protocol. That's not what it is. It's not, it wasn't intended for that. Because a value is not there, does not mean it is bad. It means it wasn't necessary for clinical operations for that particular patient. And in many cases, it could actually be meaningful. So the concepts of missingness and incompleteness, it may very well not tell a full story. It may not, because the clinician at the time who was collecting that information may not care about the full story. They care about the person's, they may care about the person's diabetes and not their psoriasis. I don't know, I'm not a clinician. Those may be very related, but maybe not. So the reason it is important is that for using EHR data for retrospective research, you do have to understand its limitations and its difference than prospective data collection in a research protocol. It is not the same thing. The value is that there's lots and lots and lots more data. The challenge is if you want a hemoglobin A1C and in your prospective data collection for a clinical trial, you would have one of those. In clinical practice, you may have 3. You may have 7. You may not have any. What do you do with that? Does that mean the data is bad? No. It means that the data is exactly what it was for clinical practice. It's just a matter of how you can use it for research and understanding that difference. The big challenge is that things are, in fact, captured in unstructured clinical text. That is, in my mind, by far the biggest challenge for secondary use of electronic health record data, is that regardless, despite the proliferation of EHR systems, there is still a good bit of content that's in unstructured clinical text. So it is a challenge. Even things as simple that you would think are very, very easily structured. Pulmonary function test. PFT equals numeric value. At UAB, that is in unstructured text. Now, it's unstructured text that we can go find relatively easily in a particular set of notes in a particular time, or if we go back 3 systems and find something that generated it, perhaps. But it's not in the electronic, it's not in Cerner and in our enterprise data warehouse in a way that I can say, give me PFTs between 7 and 12, or 97 and 102. I don't know what the right value is. So it's a challenge. So I think I've touched on most of these things. One of the areas of informatics that is probably not, that is relevant, but not very well known is in the area of terminologies and ontologies. So that's actually the specialty. That is Dr. Cimino's longtime passion, is the actual development of ontologies and vocabularies. The formalized structure for representing clinical knowledge in a structured way, how things are connected to one another. And it is a whole domain of research. The one easy example is in ICD-9 diagnoses, ICD-10 diagnoses, there's a concept called not elsewhere specified. So when you have a new version and there is a new category, things that used to be not elsewhere classified are now in a specific category of themselves. So the meaning of not elsewhere classified is different over time. That is a real challenge, and there are people who are trying to figure out how to handle that. And I'm not sure if there's a solution, but it's an interesting, exciting research area.
Can you say a few words about decision analysis? We're using machine learning to try to use predictive models to let clinicians know when they should order a test. Mm-hmm. And I know that other departments are doing the same thing. Is that sort of something you see coming down the pipe that you think would be a good place of an interface for
I do.
decision support and machine learning and clinical judgment?
I do. One of the challenges is, yes, absolutely. And where the opportunity is understanding the previous decision-making process in order to identify and determine the targets for future decision-making process. So if you see a pattern of activity, what does that really mean? If you see a patient on a medication and then not on that medication and on a different medication, or let's just forget about the first part. If you see the person on a medication and then not on that medication, what does that mean? Does that mean that medication didn't work? They had a bad reaction to it? Are they healed? What does that really mean? If they progress from one medication to another, I can conceive that that is a progression of treatment and failed first course of treatment, failed second course of treatment, successful on third course of treatment. But those relationships represented in the clinical record between condition, treatment, and response or outcome are not explicit. I mean, even in narrative text, they are most of the time explicit, but not really. And they're certainly not explicit in the data. You have no idea really that this medication was prescribed for this particular condition. You don't know if that medication was prescribed to try to impact a particular thing. You don't know if it did. You don't know if the cessation of that medication was because it was successful or because it failed. And so that is actually, I think, the way that we collect data goes a long way into how we can inform the decision analysis process and the intervention to provide information in the decision-making process. Now, that's probably a complicated example. There's a whole lot that you can do just building decision support rules. Other practical considerations is how do you differentially display those rules? How do you walk the line between showing someone something that they will eventually just learn to click through and making it meaningful? Because that's a challenge. So I've described a few times now that this is incredibly broad, and not only are there different types of analysis, different types of approaches to problems, but they're completely different application areas. So you may be doing machine learning and predictive modeling for basic molecular activity, or you could be doing it in how well the clinical education process is working for physicians to reduce implicit bias. Same technology applied in two completely different ways. And if anyone wants to talk about any particular part of these, by all means, throw something at me and we can talk. It's an incredibly broad domain. Anything of particular interest? Anything you've never heard of? So to tie into Dr. Korf's presentation on personalized medicine, the Alabama Genome Health Initiative, all of us, we're actively involved in all of those, both from a bioinformatics perspective and the sequencing, the building analytical pipelines, et cetera, but also from the clinical data perspective. So for all of us, we actually take clinical data from the electronic health record and send that to the All of Us program. And so we've built that pipeline to integrate the clinical data into that. We've actually pulled all of the genomic data, taken every single variant off of those 600 to 700,000 variant chips and put it into I2B2, so you can find people based on those particular variants. Now, I will tell you, there are 59 of them that are clinically actionable. There may be a couple hundred more that have been validated, and there's a whole boatload of them that may not mean anything, and it's a fine line between what you show people and what you don't, because many of the things that are identified are just artifacts of the sequencing and the annotation. So, but we are actively working in the sort of crossover between bioinformatics, clinical research informatics to really facilitate that integration of data across various projects, across external reference data, across both the sequencing, the annotation, the clinical data, the clinical research data, case report form data, and integrating all of that together. And we're doing that in the context of a project called UBright. So the idea is that Jim could talk about what he meant by all those individual pictures, but there's lots and lots of information and lots of people using lots of different information about the patient. And what we are intending is to sort of collate all those things together and tying information from clinical practice, integrating resources from research computing, high performance computing, reusable pipelines for annotation, as well as pulling text, structured data, and omics data together so that you can have sort of a integrated platform for reuse of tools that have been developed, a integration of resources within UAB, a delivery of data pipeline that is scalable and convenient, et cetera. So that includes pulling information into from various places, including clinical data, external reference data, and this analysis gateway here is probably understated. What that really can be is a very robust development of analytical pipelines and the ability to discover new knowledge based on all of this data over here, as well as all kinds of external reference data with the use of our high-performance computing infrastructure. So we've done that for the Alabama Genome Health Initiative. We've incorporated all their case report form data into I2B2, and we can deliver all of the omic data, all of the clinical data in a sort of unified pipeline to the researchers. The next step is to deliver it directly to the high-performance computing infrastructure. And as soon as we are allowed to put protected health information there, we'll just throw it right over there and people can do all the analysis they want. Okay. We are a couple of minutes from the end of the hour. So I can't really even summarize why this is important. Lots and lots of data, sensor data, social network data. I mean, the predictive ability for Facebook posts is probably better than most clinical practices. The ability to identify public health disasters based on satellite imaging of hospital parking lots in South America was just ridiculously cool, and it predicted it predicts outbreaks before the official reporting functions do. Probably the biggest thing that I try to highlight is the growth in the literature. This is probably even old, but I think the last thing I read, that if you read 10 journal articles a night, every night, you'd be like 7 years behind at the end of the year. It is the growth of knowledge in medicine is not such that a human being can have all that information and apply it. It's just not possible. So what that means is we have to come up with a way to deliver that information in the right way. And that is I think that is probably the biggest reason why informatics is important, because the way we've always done it is not going to work. It just can't, because no one person can know everything, and no 20 people can know everything. And I would even hazard to guess that even if you take all of UAB cannot know everything. There are things that are discovered every day, and nobody at UAB knows it until next week. So if a patient comes in today, we don't know that yet.
Why does Facebook have a better prediction algorithm than EMRs? They have better people working on their team? What's the story?
Because people, well, not necessarily that Facebook has the content in Facebook allows for a better predictive model, because people are talking about their symptoms before they are going to their healthcare provider. They're Googling for symptoms to see if they should go to CVS. So first Google and Facebook know that there's something going on, then CVS outpatient or over-the-counter medication use indicates that there's something going on, then the healthcare provider knows that there's something going on. It's just a matter of where the information lives. And the information lives in a lot of places that aren't related to episodic visits to a healthcare provider. I mean, even if you think about a person who is a heavy utilizer of a healthcare delivery organization coming in once a month, that's only 12 days a year. The rest of the year, they are still a person with conditions, with manifestations of the condition, still are generating data that represent that condition. So the more that we can get out of those small number of days a year and start observing the rest of that world, the better we'll be able to understand individual patients and populations of patients and be able to intervene. The view that we know about the patient because they've been here twice this year is just ridiculous. Sorry, my 2 cents. That's my soapbox. So I think what we need to work towards is 21st century data. It is not just narrative text on a piece of paper, or even narrative text in a PDF document that is sent from one place to another and represents a lab result that we can't do anything with now because it's in text in a PDF. So what we really need to do is work towards more actionable data, and more actionable data means more structured data, more well-represented data. I'm going to skip through that. If you have an interest in the domain of informatics, there is the association, American Medical Informatics Association, is the primary professional home for academic informatics. They have a couple of big conferences a year, one of which is focused on translational bioinformatics and clinical research informatics. They're pretty fun. They're in Washington, D.C. and San Francisco. Typically, this one's in D.C. You can learn as much as you want about informatics at one of these. So what we talked about, translating that elephant analogy, it is informatics is both the basics, building blocks, computer science, data analysis, knowledge representation. It is all the way through how you actually implement these things in clinical practice, how you affect change in the whole life cycle of biomedicine. And that is the end of my presentation. Any questions? Someone's got to have at least one question. If anyone online wants to ask questions, I probably have not done a good enough job in keeping track of the chat. But if there are questions, by all means, unmute yourself. Ask questions.
Yeah, yes. This may not be widely interesting to everybody, but so you mentioned lab results or things in the EMR that come in as pictures or scan documents, that's still discoverable information. Is there someone, because I've actually been struggling with that recently for a project we're working on. Is there somebody who can help us find that information?
The easiest way is to email i2b2support@uabmc.edu. Now the challenge is that if something exists and you find a pathway to it, we can provide it to you. Okay. Finding patients based on the things that are in narrative text in a PDF document is challenging. But and this is the case for, I'll generalize in terms of anything that is unstructured. If you are looking for all of the pathology report for a particular type and subtype for patients who have been seen in a particular place or have a particular, any other clinical characteristics. If you identify a set of 100 patients and say, I want these path reports on those 100 patients, no problem. We can do the same thing as long as we can identify what metadata is around that particular document to identify the one you want, then we can go get those and give it to you. Yes?
So I normally work at Children's, but we consult in the NICU occasionally. Oftentimes we get labs that come in as PDF, and I know in a different EMR, it's possible to enter in those lab values as external that then come up in your result with your flow sheet. Is there an ability to do that in Cyber?
That is a question probably better directed to the clinical informatics and nursing informatics folks in the health system or the CMIO. I think probably it's probably less a matter of the electronic health record capability and more a matter of the approval to enter clinical information into clinical practice and how that happens.
It's a giant pain to get them to do it. Incredible pain.
Because I'm thinking like over at Children's, half the time we get repeat lipid panels that are done at the local lab that get scanned in, and then we get named on US News and World Report or LBL's try. But it's not, it's just followed up with an outside lab.
Right. So I think that particular challenge probably has as much to do with lab certification and the approval to get things into clinical practice that a human being is reading from one place to another than it does whether you can actually build a field or a lab result spot. I mean, you can numerically result a lab test.
With Cerner, if you want to add in something like that, it has to come from Cerner, not from our HSIS people. So Cerner Corporation has to build this specific lab test name or result so that you can associate the number with it. Otherwise, it just gets external results scanned. Which is why when a new drug comes out, it takes up to a year for us to be able to order electronically. Because Cerner Corporation has to create it and then distribute it.
Any other questions? We are over time. Thank you.